Ça dépend, il y a des gens ça va rester vite. C'est pas un cours, il n'y a pas d'examen à la fin. On va faire un dernier sur la travail.
Vite. Tac, tac, tac. Bon, YouTube. Wow. Bah, ça grésille plus, non? Incroyable. YouTube, ça marche. Je suis sur Zoom. Tout? Bon, on est bon. Manque le jeu. Je sais pas si tu veux me dire de se mettre un round après. Je sais pas si je se met au premier ou au deuxième. Ouais. Sinon, je peux se mettre au premier. Si, vas-y.
Hey, Ruby, as a test, can you see the slide deck that I'm sharing?
Yeah. Right. Yeah, looks perfect.
Amazing. Right.
Issu.
test, 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 test. Pa, 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 pa. C'est bon? C'est fort ou pas là? Non, c'est bien. C'est bien? Ok, c'est bien. Yes.
Il y a des démons qui lui ont envoyé un message chez nous. Je ne sais pas s'il date de ce matin ou s'il est juste.
C'était pas de ce matin, je pense, parce que. Ouais, demandez plutôt aux gens du labo, c'est plutôt pour vous. Ça. Du coup, en attendant que le reste du jury arrive, je voudrais vous remercier d'être tous présents. J'espère que vous allez passer un bon moment. Il y a un petit livret qui est passé. Alors, j'en ai pas imprimé assez pour tout le monde. Le livret est plutôt à destination de gens qui ne sont pas forcément équipés en statistiques pour tout comprendre, et notamment contient un QR code qui amène vers des explications des slides en français, sur les premières slides. Donc, s'il y a des gens qui pensent qu'ils auront du mal avec l'anglais, n'hésitez pas à scanner ce QR code, et du coup, vous aurez accès à des explications. Le petit livret contient également des mots croisés sur le thème de la soutenance. Et honnêtement, même moi, j'arrive pas à le faire. Donc, s'il y a quelqu'un qui arrive à la fin de la présentation avec le mot croisé complet, c'est fou. Du coup, maintenant que j'ai tout le monde aussi, il y aura un petit pot qui sera après la présentation. Donc, maintenant, ça va se passer pour les gens qui ne connaissent pas. Il va y avoir d'abord la présentation. Ensuite, on va devoir tous sortir pendant que le jury fera sa délibération. Donc là, c'est à huis clos, donc vous ne rentrez pas dans la salle. Et ensuite, on pourra revenir et là, il y aura l'annonce. On l'espère positive du résultat de la présentation. Et suite à quoi, il y aura le pot. Ça implique que pendant la petite pause sur laquelle le jury délibère, on ne mange pas le pot. Ça ne se fait pas.
Je ne sais pas si tu l'as en plus.
C'est vrai. Donc,
De boire pour rien. Il y a un enfant de
attends. Avec le laser, comme ça, non? On le voit ou pas sur la photo? C'est essentiel. Magnifique. Vous aurez tout le temps dans la présentation. peux faire le site. C'est vrai? Bah, tu peux promettre dans... Ah, il y a le genre au niveau droit.
Bah, j'ai pas de droit, de toute façon.
Ok, très bien. Merci d'avoir parlé de ça.
Du coup, j'ai le PV de France.
Oui,
Ah, mais
De quoi? C'est la chemise, ça fait ça. Comment vous voulez?
c'est les garants techniques, hein?
bah ouais, mais là, il y a un truc chaud.
Il y a Rouillard.
Ruby est là, ouais.
En fait, t'es dans la réunion là. Je pense que...
Je sais pas où je suis.
Non, non,
Ah, mais comment tu la passes en...
Welcome, doctor. Thanks for coming back.
c'est vrai. C'est quoi que je puisse l'aider?
Hein?
C'est que je puisse l'aider.
Très bien.
Ah bah parfait.
Well, what's the problem? Raphaël, c'est quoi le problème?
Et Ruby est toujours là.
Hello, Ruby. Thanks for agreeing to participate.
Yeah, hi, nice to meet you. The only issue is that I don't speak
Sorry, what?
French. So I don't speak French, so I miss all the jokes you guys are laughing about. So
Oh,
the rest
don't
are
worry.
okay.
I mean, the jokes in French are not very good either.
Okay, cool.
Okay, I'll go back with the jury and thank you.
We're waiting for Rafael to give us a good. I don't know. Yeah? Okay, good.
Everything is good?
Is the jury okay?
Everyone's here?
Yes. Yeah.
People appreciate Alexander?
Yes.
Please turn on off your phones.
Good?
Yes.
All right. So welcome everyone. We'll do the presentation, I guess, in English because there's some people who don't speak French or attending. So I will act as president of this jury. So we're here to listen to the PhD defense of Louis. And the jury is composed of Sylvain Lecorf, who's professor at Sorbonne Université, Gilles Loup, who's professor at Université de Liège, Louis Kong from Singapore, Pedro Rodriguez, who is chargé de recherche at INRIA Grenoble, and we have Daniel Basserman, who was, and Luca, you're not on the...
Luca is an invitee because we are co-authors, so like officially he couldn't be part of the jury, but
Okay.
I'd like him cited as such.
Okay, and Lucas from the Donders in the Netherlands, professor in the Donders. And I think the floor is yours. So you have 45 minutes to present your work, which will be followed by the question of the jury. And the floor is yours. Oui, go ahead.
Bonjour à tous. Hello, everybody. And welcome to my PhD thesis defense entitled Bridging Simulation-Based Inference and Hierarchical Modeling Applications in Neuroscience. So the presentation will be decomposed in 7 parts. In parts 1 and 2, I will present modeling in neuroscience and application in magnetic resonance imaging that are 2 fields of application that motivate the methods that we build in this thesis. In parts 3 and 4, en fait, ça va falloir le dire. Hop là. In parts 3 and 4, I will present 2 frameworks for statistical inference, namely variational inference, or VI, and simulation-based inference, SBI. Before moving on in parts 5 and 6 onto the contributions of the thesis on the possibility to build scalable, yet both expressive and robust variational inference. In part 7, I will quickly move on to some conclusions. So part 1, modeling in neuroscience. The general question that we ask ourselves in this thesis is how to study the human brain. though the association between the brain and the mind can be dated back to ancient Egypt, Franz Joseph Gall in the 19th century first proposed a theory which is the association of specific mental faculties to specific brain regions. This theory was further supported by famous brain autopsies, such as the one performed by Paul Drucker on one aphasic patient, Victor Lebon, leading to the discovery of the area associated with speech production. Though very useful in localizing precise function at precise locations, those post-mortem studies are obviously of limited scalability. So the other issue is that they significantly alter the brain and give no insights regarding the brain activity, which is a support of cognition. Therefore, to study the brain non-invasively, we need to measure the signal that results from its electrical and metabolic activity, which is the field broadly described as neuroimaging. In this thesis, we focus ourselves on one specific brand of neuroimaging, magnetic resonance imaging, or MRI, which measures the magnetic signal that results from the brain structure and activity. Specifically, we study functional MRI, or fMRI, which measures changes in the blood oxygenation resulting from the neuronal activity. And on the other hand, diffusion MRI, or DMRI, which measures water diffusion as altered by the cellular microstructure. In this presentation, however, I will only talk about fMRI. The issue with neuroimaging is its large complexity. So we measure a very complex signal, in this case, magnetic signals, coming from an already complex object, that is to say, the human brain. To make sense of this complexity, neuroscientists oftentimes posit explanatory models that explain the complex observed signal using some simple latent parameters. Keep in mind descriptive model parameters and signal, because this is the one in which we position the applications that I'm going to describe. In the next section, I will dive deeper into what kind of brain signals and what kind of models does this presentation cover in the context of functional MRI. So the goal of functional MRI is to study the neuronal activity, but we cannot directly measure this activity, but a delayed vascular response that results from sugar consumption. And this theory is associated to the so-called blood oxygenation level dependent, or BOLD contrast. So following a neuronal activity at time 0, you will have a delayed vascular response that spans over several dozens of seconds, and that is characterized, for instance, by the time to peak at which the oxygenation in the blood is at its maximum. The issue is that this so-called hemodynamic response function, or HRF, can vary across the human brain. For instance, considering in dark a first region 1, and in light blue another region, you can see the time to peak from region 1 is larger than for region 2. In practice, this may imply that region 1 could be active before region 2, but the peak in oxygenation in region 1 will happen after region 2. Furthermore, this HRF is very difficult to identify just by observing the BOLD time series. So this is the kind of large uncertainty that we want to overcome through our methods. So fMRI, one of its primary goals is to investigate the localization of certain cognitive functions across the brain. A major hypothesis actually partitions the brain into networks that are dynamically mobilized during tasks. So network is composed of several regions that are interconnected. As a famous example, you can see here on the bottom, the Yeo-7 parcellation, that, as the name implies, partitions the brain into 7 networks associated with major functions, such as motor control or vision. The goal in here is to identify such network from the BOLD signal, which can be represented as a tensor of times number of regions times the time dimensionality. So how to define these connections across regions? One straightforward mathematical tool is to rely on correlation, which results in the connectome A of shape number of regions times number of regions. And each line in this connectome describes the connectivity of the rest of the brain compared with the seed region here in the temporal lobe, for instance. So the regions in red here are the regions that are highly correlated in the signal with the signal of the seed region, and that we therefore assume are connected to a certain extent. The two applications that I'm going to present here both revolve around connectivity. In the parcellation application, we will use such correlation-based connectivity fingerprints to map the human brain into microscopic networks, much like the Yeo-7 parcellation that I presented on the slide before. In the coupling application, we will refine correlation into a directional measure of functional connectivity. Another defining characteristic of neuroimaging is the shear scale that it entitles. Typically, data is collected across multiple subjects and multiple sessions to tackle at least two effects. First off, at the subject level, there is significant measurement noise, which means that there is uncertainty in the parameters associated with a given subject. Furthermore, there is a variability across subjects. For instance, the patterns of functional connectivity may vary from one individual to the next. In practice, this means that multiple subjects need to be considered to capture replicable population trends. To combine the information across subjects and measurements, we resort to hierarchical modeling, but this further multiplies the amount of data we have to process, multiplied by the number of subjects and sessions simultaneously considered. This means that we have to deal with very large-scale data. As an example, in the parcellation application that I presented in the previous slide, we collect connectivity fingerprints across 60,000 brain locations, 2 scanning sessions, and a mere 1,000 subjects, for a total of 7.6 billion measurements that need to take into account. So if we take a step back, we can position the 2 applications that I presented, parcellation and coupling, in terms of the signal model parameters framework. So in the parcellation application, we start from correlation-based connectivity fingerprints. We will use a hierarchical mixture model, and the goal will be to recover individual parcellations, maps of the human brain. In the coupling application, we start directly from the ball time series. We'll use a latent linear coupling model, and we'll derive directional coupling matrices. The thesis also featured a third application, the microstructure estimation application, that is not covered in this presentation. Roughly speaking, in this thesis, we aim to go from the left to the right, meaning that given the generative model, we want to know the parameters that best explain the observed signal, a problematic which is oftentimes called inference. The issue is that we provide very simple explanation for a complex reality, and this creates uncertainty. Namely, several sets of model parameters could equally well explain the observed signal. This thesis relies on statistical inference as a way to capture, quantify, and ultimately reduce this uncertainty. This means that we will not recover a single parameter value, but distribution of parameters that associate a probability to a given parameter range. From a methodological viewpoint, the problematic would therefore be, what is the distribution of the model parameters that could yield the observed signal? And can we recover such distributions even in the context of large-scale data? Oops. So if you pay particular attention to the slides, you may have noticed that 3 words were put in bold fonts and indicate 3 hurdles that are defining the imaging applications. The first one is complexity, so the complex relationship between the parameters and the signal and across the different parameters, for instance, from the ball time series to the patterns of functional connectivity. The second one is uncertainty, meaning that multiple sets of parameters can equally well explain the observed signal, so multiple patterns of connectivity for the same activity. And last but not least, the large scale, meaning that we need to combine the information across many subjects and measurements, and this through hierarchical modeling. In the next sections, I will move on to how to tackle those hurdles in statistical inference, starting with the first framework, variational inference, or VI. So VI is first and foremost a Bayesian methodology. In the Bayesian framework, every variable is conceptualized as a random variable associated with a probability distribution. As an example, the model between the signal x and the parameters theta is the joint distribution P of x and theta. Bayesian inference searches for the distribution of the parameters given the signal, also called the posterior distribution. On the right of the slide, you can see in Bayes' theorem that the posterior can be decomposed into 2 factors. On the one hand, we have the prior, which is the assumptions about the value of the parameters without even seeing the data. And on the other hand, you have the ratio of the likelihood to the evidence. Roughly speaking, in the Bayesian framework, the new assumption about the value of the parameters, that is to say the posterior, is a combination of the prior assumption reweighted by the evidence that has been presented to the experimental. In the neuroscientific context, the signal could be the region blood oxidation time series, the BOLD. The parameter could be the connectivity between regions, and thus the posterior would be the distribution of all the possible connectivities that could explain the observed time series. So the goal of a variational inference is to recover this posterior. And to do so, inference is treated as an optimization, meaning that we will define a parametric distribution, Q of theta, to approximate the target, that is to say the posterior. The weights of this approximate distribution will be optimized to minimize a distance between Q and this target. And this connection between VI and optimization is actually very fruitful because this allows us to leverage the tools from the machine learning community, namely optimizers or neural networks. As an example of a simple parametric distribution, consider a parametric Gaussian with a variable mean and covariance. The fact that variational inference relies on an approximate distribution means that the performance is capped by what we call the parametric distribution's expressivity. As an example on the top, consider trying to approximate a B-model distribution using a model target. No matter how good your optimization, you are doomed to fail meaning that your approximate distribution is not expressive enough to capture the multimodality in its target. In practice, this means that VI is a biased inference method. As indicated in this cartoon, we do not recover the actual solution to the problem, but the best approximation as part of the family of parametric distributions considered. So we want to make this cloud as large as possible to potentially capture the true solution to the problem. From a methodological standpoint, the first objective would therefore be to capture the complexity in neuroscience model. We want to build expressive distribution approximations, able to capture that complexity. So using our parametric distribution, we want to get as close as possible to the posterior. And to do so, we need to measure the distance between the two. And for this, variational inference relies on the reverse Kullback-Leibler divergence, which, roughly speaking, matches the approximate density to the target density. And it does so, and this is the important distinction, over the support of the approximate distribution. And this is an important distinction because combined with the local exploration from optimization, this makes traditional VI susceptible to a phenomenon called mode collapse, which is illustrated on the right of the slide. You can see that in the presence of a bimodal target, the approximation focuses on one single mode of this distribution, one set of solutions, thereby ignoring entire parts of the solution space. In practice, this may imply distribution uncertainty underestimation, because the span of the approximate solution is much narrower than the span of the theoretical solutions, and even in spurious results. So the second objective is that to tackle the uncertainty in neuroscience applications, we want to build robust distribution approximations that are well calibrated in their uncertainty. Last but not least, the large-scale neuroimaging prevents inference over all the subjects and the measurements at once. To circumvent this issue, VI has access to a tool called stochastic VI that allows to infer only over a subset of a hierarchical model at once. Considering the population study on the bottom, with potentially many subjects and many measurements, at a given training step, we could train over only a subset of the subjects and measures, and this subset could furthermore vary across the training algorithm, thereby controlling the amount of memory and compute that we need in our optimization. So the third objective is that to tackle the large-scale neuroscience model, we want to build scalable distribution approximations that are able to leverage tools such as stochastic variational inference. So I presented 3 hurdles in imaging, and in this section, I presented 3 desirable properties of inference, each answering to a particular hurdle. Expressivity means distributions able to approximate any target, no matter the complexity. Robustness means a parametric distribution to capture all the possible solutions to the problem and not underestimating uncertainty. And the last one is scalability, parametric distributions that can typically tackle population studies without an exploding memory or compute requirement. We'll actually separate those 3 desirable properties into 2 groups. On the one hand, we have scalability that I already talked about and is dealt with by hierarchical modeling and stochastic VI. For expressivity and robustness, we will look into the tools from another framework in statistical inference called simulation-based inference, or SBI, which is the topic of the next section. So the principle of SBI is to perform even in the absence of an explicit model that links the parameters to the signal. This typically happens in the presence of a simulator, as visible on the right. So you can feed parameters to this simulator, and it will output a signal, and the simulator can be considered roughly as a stochastic black box in this case. Crucially, you can sample from the simulator, but you cannot evaluate its associated density, meaning that once you have the signal, you cannot know what was the probability of obtaining that particular signal when given those particular parameters. As such, the branch of SBI we focus upon must rely on powerful density approximators as a means to learn surrogates to, for instance, the posterior distribution of theta given x. As a family of density approximators, we focus on normalizing flows, which stack multiple invertible transforms, represented as those blue arrows, as a ways to transform a simple distribution, so the quotient distribution at the top, into a possibly very complex distribution, such as the cross-check distribution on the bottom. In this presentation, we can consider flows as universal density approximators. So they are a good fit to tackle the expressivity requirements. But conversely, flows scale poorly to large spaces, and this makes them ill-suited for the scalability requirement. In this slide, I present the architectures in which the flows are typically used in the context of simulation-based inference. Usually, they are combined with an encoder, which is applied over the observed signal. And we put forward the duo comprised of 2 elements. On the one hand, you have an encoding that results from the application of a trainable encoder to the observed signal. And this encoding conditions a flow that will approximate the distribution of the parameters conditioned by this encoding of the signal. In this thesis, we will repurpose the flows and encoding architecture in the context of large-scale hierarchical inference. So this first section was about the architecture in SBI. Now, how are those architecture trained in practice? We start from a synthetic data set. So to do so, we sample parameters, produce parameters, feed them to the simulator to obtain synthetic signal. The signal is fed to an encoder to produce an encoding. The encoding will condition a flow, and this flow induces a distribution over the parameters. And the goal of the flow will be to maximize the density over the parameters, conditional by the encoding of the signal. So similar to VI, the goal in SBI is to minimize the distance between this flow-induced distribution and the target, which is the posterior. And to do so, we need to measure a distance. Where variational inference relied on the reverse Kullback-Leibler divergence to do so, in SBI, we use the forward KL, which is pretty similar, meaning that the expression is very symmetrical. We match the approximate density to the target density. And then again, this is the important distinction. We do so over the support of the target distribution. And this distinction is very important because contrary to its reverse counterpart, this makes the forward KL not susceptible to mode collapse. And this makes it a good fit for a robustness requirement. Conversely, as for the flows, the forward KL is ill-suited to the large scale due to the need for another requirement, which is amortization. So the reuse of a density approximator across many instances of the signal instead of just the instance of interest. So 3 hurdles in neuroimaging, 3 properties of inference, 2 of which were to be tackled using the tools from SBI. More specifically, for the expressivity requirements, we will look into the flow and encoding architecture that I will talk about in the next section. And in the section after that, I will talk about robustness and the use of the forward KL as a means to robustify inference. So in this first set of contributions, I will talk on the possibility of obtaining both scalable yet expressive variational inference. So taking a step back, to minimize the bias during inference, an approximate distribution must be as expressive as possible, making this blue cloud as large as possible. Flows are expressive, but they cannot scale up to the large dimensionality of neuroimaging hierarchical applications. For at least 2 reasons. First, they are heavily parameterized, meaning that the number of weights scales poorly with the size of the space they are applied upon, and they can take a significant training time to approximate the target distribution. In the first set of contributions, in collaboration with Alexandre Lebris, Thomas Moreau, and Damien Wasserman, we present the plate amortized VI or PAVI architecture, in which we propose to both share the parameterization and share the learning of flows across a hierarchical model. From a conceptual standpoint, the goal is to bring the expressivity of flows in the context of hierarchical modeling, starting with sharing the parameterization. In PAVI, we reuse normalizing flows to approximate the posterior distributions of parameters with the same level, focusing, for instance, on the subject level parameters. You can see that all the subjects are associated to a shared or so-called amortized conditional flow. And every subject is associated to unique encoding. The important distinction between the two is that the flow is very heavyweight, whereas the encodings, they are lightweight. This means that the flow concentrates most of the weights in the variational family, and the encodings are pretty negligible compared to that. So the fact that we share the flow across many repeated instances in the hierarchical model, in practice implies that we can much reduce the approximate distribution parameterization. In practice, PAVI unlocks flow-based inference in the large scale without an exploding memory requirement. This was about sharing the parameterization. In terms of sharing the learning, combining shared flows with stochastic variational inference, most of the weights in the approximate distributions are trained at every stochastic step. You can see here that the lightweight encodings are only trained at certain steps when the corresponding subject is visited by the algorithm. In the contrast, the heavyweight flow is trained at every step. And remember that the heavyweight flow concentrates, then again, most of the weights of the family. So we train most of the weights at most of the time. And in practice, this continual training results in dramatically faster convergence and unlocks large-scale inference using flows in the large scale, again, without an exploding computation time requirement. In this slide, I compare PAVI against a baseline built by Luca, which is cascading flows that also uses normalizing flows, but pays no particular attention to scalability. And we compare our architectures as the number of subjects in a population study from 2 to 20 to 200, moving on to light blue to dark blue histograms. In the first panel, you can see that PAVI maintains the approximation quality, meaning that the approximation of the posterior is good compared to a state-of-the-art baseline. In the second panel, we can see that PAVI controls its parameterization with a constant or almost constant number of weights as the size of the problem augments. In the third panel, we can see that PAVI converges up to 100 times faster with the same properties. So in this case, the scale is in the log domain, meaning that a difference of 1 unit implies in practice that you're 10 times faster to train. And this faster training means that we can unlock large-scale applications of inference without exploding time. For instance, we bring down the training time from literal months to hours. And this scalability allows us to apply this kind of architecture to very complex large-scale applications, such as the parcellation of the human cortex. So in this application, we inspire ourselves from existing work from Kong and Yale, and we use correlation-based connectivity fingerprints that are presented in the earlier slides of the presentation as a basis to map the individual brains of more than 1,000 subjects. So the goal is to obtain maps that very much resemble the one from the EO7 parcellation, that localize functions such as motor control or vision across the cortex. This is the first output of this application. So from the connectivity fingerprints, we want to build those individual maps. Next, we can use those individual maps as a means to predict the subject cognitive ability. So this is a very strong result when you think about it, because this means that by observing the localization of function across the cortex for an unseen subject, you can predict her or his ability to perform particularly well in the context of cognitive tests, such as vocabulary repetition or remembering previous items in a sequence when presented with series of images. So from the connectivity fingerprints to the maps to the cognitive ability. One important caveat in this application is that for a given subject, we only have access to a few measures. So the label of a given vertex may be equivocal. Said differently, for a given point on the brain, we cannot say for sure whether it belongs to the green or the red network. To capture this uncertainty and represent it, we'll use a probabilistic parcellation. So each vertex connectivity, represented as a signal X here, will determine a probability to belong to either one of 8 networks. And we obtain 2 such probability maps, one at the population level, and one for each subject that is a perturbation of the population map. So here we represent the probability for any vertex to belong to network 1 to up to 8. So as I said in the earlier slides of the presentation, this application features a massive large scale. For instance, the signal X in the connectivity fingerprints is 7.6 billion measurements, and we want to infer of a mere 400 million parameters. But thanks to its scalability, PAVI can tackle this large scale application in a fast and automatic way. So the signal is the connectivity fingerprints. The model is this hierarchical mixture model that I just presented. And the goal is to recover the subject and population level probabilistic maps. And here are the results. You can see that the population maps appear as a smooth average of the subject maps that are much noisier. At the subject level, you may even notice that certain parts of the brain are colored in white. And this is our way to represent uncertainty. For certain locations in the brain, we just don't have access to enough information to say univocally that this point belongs to either the red or the green network here. So this was the first output, obtaining probabilistic maps. Then we can use those maps to try and predict the subject's cognition. And this is visible here on the right, where we consider a set of 13 cognitive measures. And the accuracy that is represented here is the correlation of the predicted cognitive score with respect to the true score. Our architecture, PAVI, performs around 0.16, which is on par with the state-of-the-art at the time, but slightly below a revived state-of-the-art of Kong 2021, that boasts an 0.18 performance on a set of 36 measures, which is larger than the 13 measures that we consider here, and subsets of 1,200 subjects, as opposed to the 1,000 subjects that we consider here. So keep in mind those 0.16 and 0.18 numbers, because later on we'll compare those to other numbers in cognitive scores prediction. So using PAVI, we're able to bring the expressivity of flows to a large hierarchical application to obtain probabilistic maps of the human cortex. And next, we were able to use those maps to predict the cognition of subjects. This was about the expressivity and the usage of the flows and encoding architecture. In the next section, I will delve deeper into the notion of robustness and how to leverage the forward AL in the context of scalable yet robust variational inference. So the application here is to redefine functional connectivity. So remember that fMRI measures the whole time series of shape number of regions times time, from which we can derive a connectome, and the most straightforward mathematical tool to do so is to use correlation. Correlation is very straightforward to compute, very straightforward to interpret, but it lacks the finer granularity necessary to unveil the final role of individual regions as part of correlated networks. So for instance, consider on the right two different configurations for a network, featuring the same regions A, B, and C. In the first configuration, A activates C, that activates B, and in the second configuration, A activates B, that activates C. The issue is that because of this sequential activation pattern, all three regions will have correlated signal. This means that from the point of view of correlation, we effectively cannot distinguish between those two configurations. We can say that A, B, and C belong to the same network, but we cannot say who activates whom in this network. In contrast, we would like to be able to disambiguate those configurations, and this is the goal of so-called causal or effective connectivity measures that roughly aim at unveiling the final functional role of regions as part of networks. In this application, we propose to replace correlation with a different metric called directional coupling. And this was done in partnership with Luca, but the Stanford Cognitive and System Neuroscience Lab as well. The main hypothesis is that we consider the observable signal to be the convolution of some latent activation X with the HRF, represented by the letter H. So the HRF is this delayed vascular response that you have in the brain, and that can furthermore vary across regions. So this is one of the main uncertainties that we try to overcome in this application. At the latent level, X, we assume that there is a linear coupling across regions, meaning that X at times T plus 1 is equal at X at times T multiplied by a directional coupling matrix. And the shape of this matrix is number of regions times number of regions. So it roughly plays the same role as the correlation-based connectivity measure. So the signal here is the observed time series. The model is this latent linear coupling model that I presented, and the major output is this directional coupling matrix. The issue is that if you feed both signal to this generative model and use an off-the-shelf variational inference, you actually fail on that problem, and quite spectacularly. So this is the output on a synthetic example where we basically know the ground truth, which is represented by the dotted lines here. So we start with the inference of this vascular response, the HRF. So there is one different HRF for two regions, 0 and 1. In purple, you can see the distribution of solutions as outputted by a baseline based on a variational inference. And as you may notice, the set of solutions does not even include the ground truth, meaning that there's a huge bias in the inference. This was for the HRF, but now if you translate those results to the directional coupling, which is a 2x2 matrix, so number of regions times number of regions, considering the coupling from region 0 to region 1, you can see that with very strong confidence, the baseline outputs a positive coupling with a weak value from 0 to 1, where the actual coupling is negative and with a much stronger value. And now in light blue, I represent the results for a hybrid inference method. And you can see that the span of solutions is actually much larger than the ground truth belongs to one of the modes of the distribution, whereas the baseline focuses on one other mode of the distribution, thereby completely missing the ground truth. If we take a step back, this means that the naive inference method mode collapses, which is the phenomenon that I presented in the earlier slides of the presentation, in the HRF. So like it collapses in one of the modes of the distribution, and this leads to spurious coupling inference. And the culprit for this behavior is the mode-seeking reverse Kullback-Leibler divergence. So how can we circumvent this mode collapse and obtain the full set of solutions to the problem? Our solution is a hybrid inference method. Hybrid because we separate the parameters into two groups. On the one hand, we have the hyperparameters, and they represent the hard part of the inference problem, meaning that they are multimodal and they require like specific care. But if we consider plate amortization, they can be considered very low dimensional. On the other hand, you have the parameters that are completely different defining characteristics, meaning that they are relatively simple to infer if you know the hyperparameters, but they are high dimensional and they require fast inference. So for the hyperparameters, we will reproduce the same working principle as in SBI, meaning that from the generative model, we will sample a large synthetic data set. And using the data set, we will train a hyperparameter estimator using the forward KL. And remember that this forward KL prevents mode collapse. Next, we will plug this HP estimator along with the actually observable signal into a scalable reverse KL training, obtaining the parameter estimator. And because we prevented mode collapse in the HP estimator, we also prevent it in the parameter estimator. Roughly speaking, here we want to get the best of both worlds, meaning that we want to combine the robustness of the forward KL and the scalability of the reverse KL. And leveraging tools from both SBI and stochastic BI, our method can be applied to the full brain directional coupling estimation, whereas previous instances of effective connectivity measures were usually limited to dozens or so regions because they did not feature such a scalability. The first application we can do for this method is in the context of working memory. So here we try to identify the full brain directional coupling for a set of more than 700 participants. In working memory, subjects are presented with series of images and are asked to try to recall previous items or the first items in the series. A previous study led by Kai and colleagues in 2021 hypothesized the driving role of one particular region, which is the right anterior insula, as a driving hub in working memory. But this type of analysis is actually confounded potentially by the fact that we do not observe the full brain, and some unobserved regions could be responsible for the activation of observed regions. In contrast, we propose to apply the method to the full brain, and we obtain the directed outflow visible on the bottom of the slide. So the hotspots, bright colors here, represent regions that mostly drive the signal of other parts of the brain, whereas the darker regions represent regions that are mostly driven by other regions. And you can see in here that the right anterior insula appears as a hotspot in that distribution. This means that we're able to reproduce in a data-driven way a neuroscientific hypothesis that was built over a set of only 11 regions, thanks to the scalability of our hybrid method. The other application we can do in the context of task fMRI is task prediction or task classification. So we observe the coupling matrix or the correlation matrix. So in both cases, the patterns of functional connectivity. And based on that pattern, we try to guess whether the subject is currently being idle or remembering previous items of a series. So his brain is being active. As you can see in the confusion matrices on the right, using the directional coupling as a feature, we have almost perfect accuracy, 99% in this application, to be compared with the 92% obtained with correlation. So this means that under the lens of this application, full context directional coupling appears as more task-specific than vanilla correlation as used in most of the neuroscientific community. Beyond working memory applications, we can also apply our task to the resting state, so in which the subjects are asked to do nothing in particular while being under the scanner. And the goal here is to investigate the default organization of the human cortex. The first application that we try to do is to recover a macroscopic organization of the brain using this directional coupling. To do so, we cluster together highly coupled regions and progressively aggregate those until we obtain networks that span over the entirety of the cortex and that are visible on the top here. As you may see, this recovered macroscopic organization is much similar to the one that we have based on correlation, for instance, in the EO7 parcellation, with major differences located in the frontoparietal and limbic parts. So this is actually a good thing, because this means that using our hybrid method, we are able to support the known functional organization of the cortex. But now we can take any of these networks, such as the purple one here, and before correlation was only able to tell us that this network was composed of several interconnected regions, but could not say who activates whom in this network. Now, moving from correlation to directional coupling, we can disambiguate whether it's A that activates B that activates C, or A that activates C that activates B. So roughly speaking, using directional coupling, you obtain as much information as correlation, but correlation does not feature as fine an information as directional coupling. So we just saw that directional coupling allows to disambiguate network configurations. The second question that we ask is whether it yields new ways to investigate cognition. And in here, the pipeline that we use is much similar to the parcellation application. So we start from the functional connectivity, either using the correlation or the directional coupling. And using this, we try to predict, then again, the subject's cognitive ability. So by just observing the connection between the regions, we try to predict whether an unseen subject would be particularly good in lab conditions at guessing elements in a sequence or doing vocabulary picture matching. And the results are visible here. There's actually two sets of results. In orange, you have the ones using correlation, and in blue, the ones using directional coupling. And this is the same set of 13 cognitive measures that were used in the parcellation application. So you can see that using correlation, we obtain a mean performance of 0.16. And if we recall the elements from the previous slides correctly, this is quite on par with the performance that we had in the parcellation application, and slightly below the revised state-of-the-art COM 2021. Oops. But now moving on from correlation to directional coupling, you can see that we obtain 9 more points in correlation, and actually quite high correlation scores, for instance, in the context of picture matching, and even in the context of fluid intelligence, we obtain a correlation which is about 0.3, and fluid intelligence is actually much correlated to the person's IQ, for instance. So under the lens of this application, using directional coupling, we are more predictive of cognition than using vanilla correlation as used in most of the neuroscientific community. In the last part, I will quickly move on to some conclusions. So in this thesis, I presented 2 frameworks for inference. On the one hand, we have variational inference that allows to tackle large hierarchical problems thanks to structured and stochastic BI. And on the other hand, you have simulation-based inference that relies on normalizing flows with a forward KL training as a means to tackle implicit density learning. The first contributions in this work is to propose novel architectures that combine the tools from both frameworks, working towards expressive, thanks to the usage of flows, and scalable, thanks to the usage of SBI architectures. Yet, as we showed in the second part of the presentation, expressivity is not all. You can have the most expressive distribution approximation possible, but if the latter mode collapses, you will obtain spurious results. In the second part, we illustrated the potential for hybrid training methods as a means to make VI more robust by combining the tools from both the forward and the reverse KL. Beyond those methodological contributions, we illustrated the applicability of these techniques to concrete, complex, high-dimensional neuroscience problems, obtaining correlation-based individual probabilistic parcellations, refining correlation into directional coupling, as well as reducing uncertainty in microstructure estimation, which was an application that I did not present in here. And as a set of closing words, I restate the last sentence from the thesis document, which is that we believe in the capabilities of these techniques to foster model-driven, interpretable, and computationally efficient neuroscience. And with that, I thank you for your attention. So I'd like to say for the people present that now is an okay time if you want to delve into other activities, because usually it's harder to follow. Autant dire.
There are a few brave ones that are staying. So, looking forward to your answers. All right, thank you very much, Louis, for the presentation. We'll switch to questions, and so the tradition is that we have first the reviewers asking the first set of questions, and so I would suggest Gilles Luc from University of Liège to ask these questions first.
Just a check, Ruby, can you hear well the questions as they are pronounced in the microphone?
Okay.
Okay, great.
All right, so I'll start then. So first of all, I would like to congratulate you on the quality of your work. I've read your thesis really with great interest. And I must say I was quite impressed how you managed to scale simulation-based inference to such large problems, like millions of variables to infer. It's really impressive. So I would start, nevertheless, with the basics. So usually I was a bit puzzled with some of the pages that you've written in the background. And so I would like just to start the discussion with some clarification. So could you, for us, clarify the distinction that you make between frequentist and Bayesian inference? Or rather, between frequentist and the Bayesian statistics?
Okay. Alors, le grand jeu dans ces trucs-là, c'est vraiment de deviner les questions qui vont être un peu posées. Donc, j'avais essayé de préparer une slide.
But if you read the report, you cheat.
You had a negative report.
So
Well prepared.
No, but
en fait, so first I would start with a bit of context because I think what I did badly in the thesis was not to position the type of public I was trying to address to.
so specifically, there's quite a bit of a fuss in the neuroscience community of a switch from frequentist to Bayesian. And my point in trying to disentangle the two were to show that I did consider those two as complementary tools and not necessarily to be opposed, and that they answer to different questions using different tools. As a means to illustrate that, I just wanted to say that, so in the Bayesian framework, I think one of the biggest differences is that we want to position everything as a distribution, even the recovered distributions, and a necessary tool to do so is to impose priors, for instance. And one of the often critics, for instance, in the neuroscience community, is that sometimes you don't know what are those priors. And also in the neuroscientific community, then again, the Bayesian methodology were systematically associated to those inference methods, such as MCMC, to obtain the full set of solutions, and those were considered as poorly suited to the large scale and things like that. So it's not necessarily the frequentist and Bayesian theories that they were trying to disentangle, but the usage of them in the neuroscience community. So typically, in the neuroscience, there were either parameter estimation applications by the means of obtaining the posterior distribution that were opposed to frequentist hypothesis testing with the p-value. And actually, you can do both with both, as this table underlines. So you can do hypothesis testing using Bayes vector, and you can obtain maximum likelihood estimates, even with confidence intervals, by comparing the p-values that you obtain for frequentist optimization, which may, in certain cases, even be more computationally effective. And to be perfectly honest, also in the context of large-scale applications, the weight of your evidence is usually so strong that you tend to completely crush what you do with your prior, except for certain particular parameters. So in this regime, I think that frequentist and Bayesian viewpoints actually converge. Because no matter how informative is your prior, you will obtain more or less the same set of frequentist solutions. So I agree with your remarks. For instance, my typical public was like a neuroscientific that would stumble upon this and would have known the inquisition of Bayesian methodologies in the neuroscience community and would like to precisely understand what we try to do here. So compared to most of this, I would say that what we do is estimation with parametric models. And this is opposed to hypothesis testing in a univariate context, as done in most of the task fMRI, for instance, which is testing every voxel independently and doing a statistical test with multiple comparison corrections.
Okay. So I agree with what you say, but I think it's still a shortcut to say that frequentist is only for like this simple test. If you look, for example, at particle physics, I've worked with them quite a lot.
Yeah.
And we even started SBI for frequentist analysis in particle physics. They have population models, they have hierarchical models.
Yeah. And
it's all fine.
Yeah, so I completely agree that we can do parameter estimation in the frequentist domain. And it even makes sense under a large scale.
Okay, so then I have a question about what you've worked on and contributed, plate amortized variational inference. And I just have like a very naive question. So at the end of the day, you propose two variations of the same algorithm, as far as I understand, PVF for the free parameterization and E for the encoding. Which one would you recommend using? So if you have to advise your colleague, which one would you recommend?
I would recommend PAVF if you can afford the memory. So like the fact is that PAVF still scales linearly, but very like lightweight scaling with the number of weights. Number of subjects. But in practice, I oftentimes found it was more effective at training faster and easier to hyperparameterize. So if you can afford it, I would use PAVF. Also from a theoretical standpoint, PAVI-F is unbiased, whereas PAVI-E can technically be biased. So we tried to study the regime in which this bias could be observed, which we quite did not find. But I would even recommend always testing PAVI-F first before moving on to PAVI-E to assess whether there is a shift from one to the other.
So I was actually interested by what you said then, like the fact that PAVI-E might be biased. So can you tell us about a bit more about that? So in what sense? So are you basically making assumptions about the posterior approximation that you are building?
So in the I can basically restate the elements from the supplemental. So PAVI-E relies on an encoder, so applied to a set of summary statistics at the lower level to obtain the summary statistics at the higher level. And the issue is that, do you want the equation or do you prefer one? Okay. The issue is that we basically considered that the statistics, by observing the full population, are well approximated by computing statistics over subsets of the population, meaning that in expectation, the assumption is that like if you consider a room of like 30 people and you take 10, 10, 10, 10, 10, in expectation, you obtain, so the expectation of your statistics computed over a subset of your population is in expectation equal to the statistics computed over the entirety. So for certain type of statistics, such as the mean, it is true. But for other type of high-level statistics, it may not be the case. For instance, in the paper, we actually tested higher level statistics to try to break PAVI-E into like having not the same results for subsets. So in cases where we expect that at the higher level, the relationship is much higher order, we could typically break PAVI-E. But to be honest, most of the hierarchical models usually functions by the mean and like perturbations of the mean or things like that. So in this regime, I would expect that PAVI-E would behave as intended.
Okay, okay. And then a last but not the least of the questions. So with my previous question, basically you have a choice between PAVI-E or PAVI-F, and there are plenty of other choices you have to make when you run SBI, what you propose. And then you have shown us very impressive maps of the brains that correspond to realizations out of the posterior. My question is simple then, how do you know that you are doing a good job at estimating this posterior in the first place?
This is a very good question. Actually, one we stumbled upon.
Because you had this third example where you said, look, if we knew the ground truth, it would be biased. And of course, this is convincing when you say it like this, but in practice, you don't know the ground truth.
Yeah.
So how do you know yourself?
This is a big question, and I think this is oftentimes a question in parametric modelization in general. So I would distinguish two different types of validation. The ones are the in-house validation and the external validation. So
just to be, I'm not talking about validating the model, the neuroscience model. I'm validating
the results.
I'm asking
the stability, for instance.
Like, is your approximation of the posterior faithful? Is it close to the actual posterior, the ground truth posterior that you are trying to estimate in the first place?
Okay, so I did not understand the question the first time. So good that you. The thing is that ultimately we don't know, and the goal with PAVI is to make as expressive a distribution approximation as possible, so that in most of the cases you will not suffer from bias. That being said, for instance, I did experiments in toy dimensions with the same generative model, and I looked at the calibration of the estimation, whereas it was containing the actual ground truth, whether it was true to the, and also build up from Gaussian mixture models in general up to this application by stacking like multiple layers and like adding more degrees of freedom. And I was quite convinced in those synthetic experiments that the approximation was not coarse. So given that we have the same conditional dependencies and the same family of approximators, the leap of faith was then that we would obtain good performance in the full scale. But our means of trying to validate this is also to try to obtain external input, for instance, to use downstream analysis. And we down the line try to much more guide whether we were doing a good job by the fact that if we're doing a good job, we're capturing biological signal and the downstream task performance was increasing and things like that. So, which is then again not saying that you don't have any bias, but at least you really capturing something that makes sense from the biology.
Because I was actually a bit surprised to notice, or maybe I've missed that, but simple like posterior predictive checks.
I did not like compute exactly, but I represented those in my. Maybe what is actually lacking in the thesis is that there's a bunch of validation that I did, which are the bottom of the iceberg, and I just represent the tip of it. For instance, it's also the case for downstream tasks. There were remarks or like, did you do any hyperparameter sweep? Did you try to test a different classifier or a different regressor? Which I did, but did not represent because I considered it to be a bit of a too much fuss, which was in retrospect a mistake. But PVC, I did them and a lot of visual examples and in Gaussian mixtures as well.
Okay. And then last one was still related to this problem. So at the end, you get a posterior over millions of dimensions of variables. So it's already, I mean, it's already an achievement just to get something which makes sense and which seems to make sense also biologically, but it seems also very difficult to validate that this posterior is close to being correct. So how would you do, can you foresee some methodological contribution towards the validation of these posteriors at search/Hierarchical? Could you, for example, try to adapt coverage tests to search large dimensions?
Yeah, I mean, the best would be for me to take a ground truth from the generative model and do any kind of coverage testing or PPC because we can sample from the generative model that we apply. But the issue is, and I think this is the bigger issue, is that there's a...
but you would need the whole thing to be amortized then.
Yeah, I suppose. But I think the bigger problem is model misspecification also in this case. Is this that we can sample in a lot of different synthetic scenarios, but the fact that we apply the method on a point that most likely does not come from the distribution means that ultimately we will never be sure. So down the line, I think that I've been mostly convinced throughout the thesis by validation in controllable small scale, even with amortization, and consider that as a good proxy to the large scale. But I do consider that validation, even synthetic in the large scale, is very hard, if not impossible.
But do you think we could actually try to validate or do the calibration or the coverage test using the same hypothesis, the fact that your model is hierarchical? Could we also basically break down the coverage test in small blocks and try to validate some of the parts?
So the assumption is that if it is well calibrated and has good coverage on the subset, it has good coverage over the entirety. Yeah, that seems sensible, but the issue is that depending on the statistics, the fact that the statistic is good over a subset does not necessarily mean that the expectation will be good, and that's the bigger problem. So I would be quite touchy about this. For simple statistic, yes, but not all, I guess.
Right. Thank you.
Right. Thank you very much. So I'll leave the floor to Sylvain Lecorf for the next round of questions.
Thank you. So I'd like first to thank Louis for your presentation. It was really clear. And I'd like to say that I was a bit impressed by the fact that you set aside all the technicalities that are present in your manuscript to just focus on the context, data, applications, and what you could get from your experience. So it made it very clear. And as a neophyte in the neuroscience application, I got many things that I did not really understood in the manuscript, not because it was badly written, but because it's really complex. So you managed to provide a message which was really clear in a context which is not so easy. So congratulations for this and for all your hard work, because in your manuscript you have got a lot of technicalities and implementation details which are really hard to follow, but you need to detail them to make it complete and to understand why it works. And I'd like to say that it was not so hard to read, while it would have been if you didn't spend a lot of time writing it clearly for us. So thank you for that. First, maybe a naive question on the amortization that you use in all your implementation. You said that the amortization comes from the fact that you share the conditional flow and the encoding process hierarchically. But doing so, you also lose a bit of expressivity because you share parameters at each level. So again, there is some kind of a trade-off between a complex model for expressivity and computational time. What is the take-home message on this part? So you made a few choices.
Yeah. Yes, we would lose expressivity only if, I would say, the size of the encodings was below the size of sufficient statistics from a theoretical standpoint. So if you consider that you had a perfect conditional density approximator, and that you could capture all the sufficient statistics in the encodings, you could assume that under perfect optimization and perfect approximation capabilities, you could have the perfect posterior for every subset. So I think the real idea of PAVI is to just have the necessary information. And so typically you would like your encodings to just encode the sufficient statistics to be as lightweight as possible. And you would rely on the conditional normalizing flow to then translate the sufficient statistics into the actual posterior. In this regime, I guess that you wouldn't lose any inclusivity compared to having one single flow per subset. That being said, in practice, so in the supplemental from PAVI, we try to talk about the decomposition of the error in the optimization error, the approximation error, and things like that. So the first applications that I was doing were actually using one flow per subject, for instance. And the performance was not that good. And I think because this is kind of a hard optimization problem. And the fact that we simplify it by sharing the flows across many repeated instances, so this amortization may simplify the optimization problem. So in practice, I think that the fact that we reuse this may reduce our expressivity, and so may increase the error from a theoretical standpoint. But the fact that we simplify the optimization problem may makes us gain more than we lose. I don't know if that's makes sense or
that makes sense, but I mean that there is something you need to really take care of in the encoding process. So there is
this encoding size is critical. Yeah.
In your all your implementation, you use transformer, set transformer, if I'm not wrong, but you end up saying that it's really low parameterized, which I was a bit I don't know, amazed, because I would expect this kind of encoding process to be very over parameterized. So
yeah, no, so I think we should try to be over parameterized. For the set transformer, it's not costly to be over parameterized because it's shared. In the encoding, I would say that you would need to be above the sufficient statistics and over parameterized as well, because in practice, in the synthetic experiment, this increased the conversion speed. But the possibility to be lightly parameterized should not go beyond the size of the sufficient statistics. Otherwise, this is when things would break. But in my experiments, I try to always over-parameterize the set transformer and maximize the size of the encodings with respect to the available memory. By that, hoping that we are above the sufficient statistics. Okay,
thank you. And so between your two implementation, ADAVI and PAVI, the main difference is in the way you express the posterior, the variational posterior. In one case, it's a field approximation, and in the other case, you've got some kind of structured variational posterior. And the choice, so it's I guess, easy to understand why it's better to have structured variational law. But in your paper, PV, you say that you use for the decomposition which replicates the prior decomposition, which is a choice. I guess you don't have any reason to do this. So saying that you want to do something else than in-field is really easy. But once you've said this, of course, it's really hard to choose a good way to decompose the posterior. Why did you make this choice? And do you have other possibilities in mind?
I think this is the most interesting avenue in this kind of research to me is this idea of what conditional dependencies do you want to take into account in your posterior. To be perfectly honest, I took this as a first step towards, and this was a historical step, like moving from like the classical mean-field VI. The first step was to reproduce the conditional dependencies. And I think they are good to approximate most of the dependencies that we actually have in posterior, except in cases with colliders and stuff like that. So like you actually covered most of what you need by just relying on and that's the first element. So I think it's a good first step to make. The second element is that way we can also leverage the generative model itself. So we can use the distribution from the generative model as priors or as a base distribution that we can push forward through the flows, which means that the underlying assumption is that if you have the values of the upper level in the posterior, you feed them to your generative model, you obtain a distribution. This is a good starting point, meaning that it's not too far from the actual posterior, and you have to minimally change that one through the flow. This was the underlying assumption as well. That being said, I think if I were to take this a step further, I would try to look into a faithful inversion, such as the papers from Christian Rouillard, which is that from a theoretical standpoint, given a graph, you can compute the actual set of conditional dependencies that you would need to not have any bias. The issue is how to combine this with a stochastic variational inference, because typically in a hierarchical model, as represented here, this implies conditional dependencies across different branches of the models. And this, in practice, means that even if you faithfully inverse the graph template, you do not faithfully inverse the ground graph, and you still have a bias. But I think that if I were to do it again, I would not take the prior. I would do a faithful inversion of the template, for instance, and use that as a set of conditional dependencies that would be, I think, closer to the set of dependencies that we need. And it's actually like something that they then did in the So then with the Macker group, they combine this with, for instance, diffusion models. They are proposing to do the masking of a diffusion model in the attention based on the set of conditional dependencies that you can obtain. So like they did this for normalizing flow and then diffusion models. And actually they go like really across the board in the 2024 all-in-one simulation-based inference paper, in which they do dynamic masking based on what you want to amortize or not. And based on this condition and attention architecture. So I think that just consider that you can put any mask and have your architecture be parameterized by this mask is the way I would try to do things.
Okay, I'm not sure I would go this far.
Okay.
With our solution, it's really complex and many things are intertwined there. So, but I agree with you that this is, I guess, a really good research perspective, because in more theoretically oriented work, it has been proved recently that adding structure in your variational law provides identifiability and makes you able to prove Minimax rates of conversion. So you can prove things on your variational posterior easily if you've got structure in it. So this is pretty nice. And in simpler models like hidden Markov models, you know that there is a structure in your conditional distribution, which is not the one which is present in the prior. So you need to take care on which one you're going to choose to parameterize your posterior. So this is.
I think in general, the advantage of the architecture in PAVI is that you can take the value from any random variable that you have sampled, and then you can feed this to condition the flow of another random variable, meaning that it is compatible with, even in the stochastic regime, with any set of dependencies that you do over the template. So here we did the prior, but it could be much more effective to do a faithful inversion.
So maybe one very quick question that I need to ask, I guess. In some of the toy models you used to illustrate that your implementations were good, you, except if I missed it, you don't compare it to some MCMC procedures, which would target sampling from the exact posterior. So in cases when you know the ground truth, I would have done this for obvious reason.
Yeah, I regret. I have many regrets.
I guess it would be nice of course, this won't help you a lot for the neuroscience application, but at least in toy models, this will help you understand if your parameterization are good, if you recover the modes of your posterior. If this was a lack of time.
This was a lack of time, yeah. So I took the pragmatic choice. I knew that down the line I wanted to do super large scale applications, so I focused solely on BI. And yeah, I agree with you that to validate in the synthetic regime, MCMC would be great. As a matter of fact, also in the last parts of the PhD documents, I try to touch upon the possibility to combine both frameworks because I very much like the Nutra HMC-like architecture that try to use flows as part of MCMC kernels. So I think the usage of a Markov chain as a means to provide samples to train using the forward KL, even in the non-amortized regime, I think is also a very promising avenue of research. So beyond validation in synthetic cases, I think that MCMC offers an avenue to leverage EFKL even without amortization. So.
Yeah, it's true, but even in papers which pretend to scale MCMC to large dimension, you wouldn't target this kind of complexity regarding your neuroscience application. So at least for an application, from an application point of view, it's obvious that you didn't use MCMC in your most sophisticated experiments. Okay, so maybe if you can go to slide around 74, 75, just one or two quick questions on the applications. Yes, so you provide the elbow to assess the performance of the algorithm with respect here to the complexity of the problem. Did you have a look at other metrics? Just
yeah, for instance, the Wasserstein distance with the theoretical posterior in this example.
And you never tried to have a look at what happened in the uncoded space to see using different, if you use Adavi or PAVI, when you have a look at the uncoded data, if you use any projection to have a look in 2-dimension or 3-dimension, if you recover modes in one case and not in the other, or
no? As a matter of fact, I think that working on the encoding space and bending space as is, trying to do transfer learning and things like that, is actually something that we want to do as an extension of the work with Alexandre Lebris. I never actually tried to. So what exactly would you? So looking at the encoding space, we would try to predict it non-linearly and see if we obtain the same data.
So in cases, I don't remember if in this case it's not the toy example, so you don't know the ground truth, but in cases where you know the truth, you could have a look at what if you recover the posterior distribution well, so the modes or whatever, which is not given in the yellow.
Yeah. So that using the encoding, we would be able to obtain, for instance, the mean and the covariance, like
for
we
instance,
know,
or any
no
conditional expectation of quantity which
The thing is that I think we would need to rely on a third block because even if the information is encoded in the encoding, it's not necessarily clear. So I guess we would need to at least rely on something that ensures that the information is present, but because the encoding most likely would not be the mean, and then the next element is the covariance, right? It would be present here, but not necessarily in that order or in that form. So
even to assess what happens in one encoded space and in another, if you go from ADAV to PAVI, or just see what is the impact of your choice in the variational family to what you recover in the encoded space, because then you're going to use it for many applications. So.
No, the short answer is no.
It was just a curiosity question. And maybe the last one in slide 81, and it's also in the manuscript, but I did not understand. You talk about uncertainty and quantification, which is one of the use you can make of partial inference, but I don't really understand what it means. You said that the white areas are the one which is the most uncertainty, but I don't know what it is.
So in the logit space, you can either have a logit which is much stronger, logit for the softmax, compared to the other ones. And in this case, if a logit was much stronger, we consider that it was a confident association with one of the networks. In certain cases, the logits were more evened out. In this case, we considered that the association was less sure because depending on the sampling, you could either have the association to one network or the other.
You mean that you had a threshold?
I literally translated the amplitude in the maximum logit into a color scheme. So there's a one-to-one mapping from
logit, then it goes into a softmax function, so it's invariant by
yeah, and then this probability. So if probability is 1, you have an alpha of 1 on your primary color.
Okay.
And if the probability is much lower, the transparency goes down and the colors are mixed together, and you end up white.
Okay, and just and then in there, just sampling from your flow. For you've got your logit, you use softmax, and then you sample many times from this flow and just assess the viability of what you sample.
Or depending on the parameterization. In the later applications, we actually thought this. So in here, the distribution is approximated by a flow, so it can possibly be multimodal. The later ones, we thought maybe this was overkill, and that to represent the variability in what is already a probability was a bit too much. So we actually ended up doing a Gaussian. And so in here, we just took the mean because this was representing the expectation of the logit. Okay. This is not the case here. So this is based on the things you described with the normalizing flow. And in later applications, we just rely to. But the advantage is that we have access to a notion of uncertainty, also with the spread of the logit. So you can know how much does your probability vary, and you can then again translate that into a notion of uncertainty. But we most likely collapse to the two summary statistics of variance and mean. Otherwise, it's just too complicated to interpret because the higher order statistics of what is already a logit is.
Yeah, just to understand what you implemented. Yeah, thank you.
All right. So I propose now we switch to the examiners and from the person who are the furthest to the closest. So Ruby, can you hear us?
Yeah.
All right. So how about you to take the floor for your questions?
Right. So I think my question will be very different from the other examiners, since my expertise is mainly focusing on neuroscience part, even though I was also doing algorithm methods. So I can tell Luis is more engineering background, and I assume this defense won't be focusing too much on explaining the neuroscientific findings. So my question it will be given that the motivation of this AVI model or a PAVM model is to solving the problem for large scale parameters or measurements, my question is like, why not use the traditional hierarchical Bayesian model using some algorithm like EM to solve it instead of like going to such a complicated models? Because given that this work kind of relate to my previous work, so I have to say that actually we also tried the AVE at the beginning. Not the beginning of my project. It's like one of our students is actually doing that. So one question we ask ourselves is like, why we want to use that? And we think one key reason is probably because it can be faster. Because my model is actually multi-layer things, and by using the variational EM, it's actually super slow for training. So we hope that maybe use the ABI, it can like proceed everything faster and then produce a reliable like a model. People can use it to infer individualized translations for their own data.
Yeah.
And unfortunately, what we find is that the resulting time is actually comparable to the traditional model. I'd like to see like what your comments on that.
So to European, I can say like, so we had an exchange of emails at the very start of your thesis. Then again, thank you for sending some pre-processed data at the time. I think you really like touching what is at the core of the motivation of the methodology that we're building here. So two different points. The first one is that it's very complicated. Yeah, I agree. But the goal is to make it complicated for the methods people, but not for the application people. And the goal down the line of automatic BI, I think, is that you could simply feed your generative model, whatever it may be, and you obtain automatically a posterior that makes sense and will train fast over your problem. So the idea is that compared to traditional expectation maximization, where you have to do derivations by hand to obtain your optimization routine to maximize your basically like approximate your target. Here you don't need any theoretical legwork. You just feed the generative model and you obtain the solution and train. And so down the line, the idea would be that people that would not be equipped at all, they may not understand the method, but they couldn't have done the EM derivations, and so they don't need to. So this is kind of the use case for the neuroscientist.
Right.
Yeah, and the other point is the connection between VI and EM, and I think it's very large. As a matter of fact, if you just do an estimation of the maximum likelihood value for certain parameters. So you just obtain a scalar and not a full distribution, whereas you obtain full distribution for others, you kind of like converge to the EM setting, right? By differentiating your parameters into two groups. And as a matter of fact, I think if you rely on optimization and not a by hand derived optimization routine, but automatic differentiation VI, and you decide to approximate certain parameters using a Dirac distribution, so like a maximum likelihood value, you obtain an optimization, automatic differentiation version of EM, in a sense. That has the same convergence properties. You will most likely fall into a local minimum of the loss. That much depends on your initialization, but that does not require hand derivations. As a matter of fact, in the software implementations of PAVI, we give that leeway to the experimenter. Meaning that you can select certain parameters where you want to have access to the full distribution of your solutions, and certain parameters you're just interested, for instance, a variance parameter or even a log bit, you're just interested in the expectation of it. That being said, I think that so the big difference compared to EM is then what parameters do you want to have the full distribution for, which is much more computationally costly, and what operators you accept to have only at scalars output. So of course it depends. But certain parameters, just by having a distribution, even a simple approximation such as a Gaussian one, you already have a notion of uncertainty by estimating the variance in your distribution. And I think this is a good output to have. For instance, I know in your papers that you spend a lot of time trying to validate the parcellations by seeing the stability across different subsets of subjects or that kind of things. meaning that down the line, we know that the algorithm is not going to fall into the same solution each and every time. And instead of reiterating many times the algorithm, having an estimate of the uncertainty through the variance of the posterior is already something that is good in the sense that it allows you to know whether you will be super unstable depending on your sample of subjects and connectivity or your preprocessing or not. So the major added value from VI to an EM on those parameters is this, I think. Estimation of the uncertainty in the mode you are falling into. I don't know if that makes sense for you or.
Right. So I think it's actually, how can I say, like, it's nice to see the application for like usage scenario. I guess it really depends on where like the ultimate goal of this project is or your paper is. Like, I mean, if the ultimate goal is to convince the like neuroscientists or anatomists, like this is like our postulation result is reasonable, then it's the other story. Because as I said, like to convince, then like you need to do so many validations, and that's a lot of work. And trust me, you don't want to go through that nightmare. But
No,
if it's more like
I think the goal is to position ourselves as methods.
Right.
Basically tell the community, look, you can reproduce groundbreaking work in the past using our method without the hassle of going through hand derivations yourself. So next, you can do your own model, use the same method, and we look to the neuroscientific community to propose models that are automatically inferred upon to get the insights.
Right. So another my question for the work is like I may miss that part. So did you have like training test split, or for this algorithm you are inputting like 1,000 data in parallel, and then you produce one group template together with like 1,000 different individualized translations, or you like train the model and then you have this model and then you apply to like a separate set to get the translations results and etc.
So two steps in that answer. Sorry, we didn't multi-step answer, but first there's an optation of the parcellation, and then there's the downstream task. In
Right.
the optation of the parcellation, we applied the hierarchical model over the entirety of the population at once. And then we did trend-test split in the downstream task, which means that we took care of the potential leakage in the downstream task, but there is leakage in the inference because, and I think this is the same as what you did in your papers, that you, for instance, you were fitting your hierarchical model once of the whole population and then doing a trend-test split. The thing is that then you statistically entangle your parcellations, and so there is leakage in that regime. So the thing we're trying to move to is that we would infer over subsets of the populations. So for instance, if you have 1,000 subjects, you do your hierarchical inference over 900 subjects. You keep the 100 others untouched. Then you can do the inference over those 100, but by keeping the posteriors from your original population as a prior, so that you can transfer the information in terms of hierarchical modeling from the train set to the test set, but without any leakage of the test to the train. And then you can do your downstream task here. That would be the better version, but that requires doing bootstraps of data and many hierarchical inferences, as you may have stumbled upon yourself in your experiments.
Right. So So for your prediction, for behavior prediction part, are you also using the like a similar work as my previous work, or you have a different way, like a cross-validation or linear regression or other prediction models?
Yeah. So as I recall in the your original paper, so the MS HPM, you were using a DICE kernel to do your regression model?
Not exactly DICE kernel. It's more like you can consider it as a DICE kernel, similarity kernel, yeah.
Yeah, similar, but down the line using a kernel method. So the underlying assumption is that if under your kernel your parcellation is similar, you should have similar scores.
Right.
So we tried to do that, and we compared it with several other possibilities. Actually, I can show you those. So those results are from papers in preparation with Alexandre Lebris and Demian. Oh no, pas du tout. Okay, so in here, you basically can see the regression curves.
Right.
And we're using different features. And you can see, for instance, we're using two different stuff, either the connectivity fingerprints, so the mu, or the topology, so the theta. So the logits of the repartition, and we obtain roughly similar performance, but slightly better for topology down the line.
Right.
And for this, we basically feed those directly to linear regression. So actually, it's a ridge where we do the hyperparameter setting for the alpha so it's the CV ridge.
Right. And there's also one like okay, I see. So okay, actually that leads to another question. It's like, like normally when we do this kind of thing, it's always the one key question is always like, are all these comparable? And this plot this plot makes sense to me. However, like I remember you have a table showing the different prediction accuracy across different methods. That is a bit like unclear to me whether like you extracting number from every paper individually, or it's like you rerun everything using the same framework for your own prediction, and then you got the numbers.
So I use the same, basically I extracted the results from your 2018 paper. I use the same HCP population, not necessarily with the exact same bootstrap because I didn't have a
No,
test.
yeah, the bootstrap part is fine. The issue more is like for example, mine was like 17 networks. It's gonna hugely like impact a lot of the results. But
Yeah.
I don't think, I think that same with other people's work. Yeah. The number of networks.
Yeah, I did not reproduce the pipelines. I used existing results under the basis that the main point of comparison was the MSHBN, because we used mostly the same generative model. And the idea was, can we compare to the MSHBN? And there, the underlying assumption is that we were taking the same model, just a different inference method.
But the number of networks is different,
The number is different too.
Yeah, that's gonna change results a lot. If it's like for paper publication, you may consider to be consistent, or you can, I don't think you want to run my algorithm to generate like 8 network parcellation. But I mean, from the fairness perspective, you might want to be consistent. Yes.
Yes. In the number of networks. So as a matter of fact, in the later. Oh yeah. I think this is with 17 networks, which is similar to yours. So the plot in itself is like the impact of transfer learning from the HCP population to the CAM-CAM population. And I think those maps and the performance that we obtain in those plots, they're on 17, and therefore they are much more comparable to what you do.
I don't know, you will find it helpful or not. Like we do notice similar, like this noisy small patches pattern for when we try the ABI ourselves, the amortized variational inference ourselves. And interestingly, we don't see this problem when we use the group average clean data. So which means if we use the group average clean data to estimate the brain parcellation at a group level for like using the YO 2011 algorithm and also use the amortized variational inference, the results are identical. However, if we use a very noisy individual data, they will give slightly different results. I don't know why. Yes, I don't know why. Just like something.
Well, my interpretation is that it's a highly non-convex or something.
So, yeah.
It's sensitive to your initialization, and it's sensitive to your optimization if your data is very noisy. And so the fact that you denoise your data probably first like simplifies this landscape, so you tend maybe to smooth it, and maybe minimizes also like the model mean specification. So like I would say it makes sense if you apply it on noisy data.
It seems like
That
the
we-
inference will be very tricky. It will jump into some local minima very fastly. Like somehow, yeah. As you said, it depends on the thing, but we don't know how to resolve that. That's why we gave out that project, by the way. So anyway.
That being said, because of it's not optimizing simple scalar, but for instance, a Gaussian distribution, I would expect that the variational inference method would be less sensitive to very tiny because of the noise that
Yes.
because you're fitting a whole distribution and not a single point. So like you're less likely, I think, to get stuck because whenever you sample from your variational, you always a bit around.
Right.
So that what you say is surprising because I would expect the AVI to be less noisy in the context of noisy data.
Yeah, I don't know. Like we suspect is maybe because when we run variational EM, like there are these iterations actually kind of like smoothing the results a bit. But for AVI, when it's estimated, when it finishes the inference, it will like produce a result without much fine tuning. But for the variational inference, there's some actually like during the iteration is more like a fine tuning the results and becoming like smoother. Maybe I don't know. Yeah.
But down the line, I think EM and variational inference, especially in the context of automatic differentiation, tend to become one and the same. And I think it's actually very fruitful to try and combine both by selecting the parameters for which you don't want the uncertainty and are fine with a scalar estimate versus the rest.
Right.
And I just jumped on the occasion because kind of proud of this, but this is one of the additional results that we had done by Alexandre. And here, this is the missing thing from this manuscript, meaning that we were able to transfer oscillations from HCP to CAM-CAM in that regime, showing that even over a small set of subjects, you obtain much faster a good performance in cognitive course prediction. So this is what I was hinting by the population split, so you do your hierarchical inference on a certain set of subjects, and then you transfer those representation to maybe another data set.
Right.
Use this for inference. This is the kind of results you can obtain even on different data sets.
Yeah, that's what that's what we do. We actually train the model using HCP dataset, and then the pre-trained model is released, so people can just use it to apply on other extra datasets.
Yeah.
So yeah, and actually the test step, so we gave out this like amortized variational inference project is also because we noticed that when the model is trained for testing part, actually it's super fast already. It's like around 3 minutes. The most of the timing is actually reading the MRI data itself. So that's why we kind of like, oh, maybe it's gonna be super hard for us to beat our own like a variational EM algorithms. So yeah, anyway.
Yeah, I can believe that, especially if you left the upper parts of the hierarchical model untouched, just optimizing for one subject or a dozen subjects is not so long.
Right.
And you benefit much from the smoothing effect from your population.
Right. Okay. Actually, I do have more questions, but I don't think like it's appropriate here because it's too like a neuroscientific things. I don't think like it's suitable.
I know it's a bit of a stretch to try and combine statisticians on the one hand and neuroscientists on the other.
Yeah, it's very different. So I will just like I think that's all. Yeah.
Thanks.
Thank you. Yeah.
Thank you very much. So I suggest to give the floor to Luca and William to ask his question.
Thank you. So congratulations. I really appreciated your work. I was particularly impressed about your PAVI paper because I think your way of extending amortization to this kind of more fine-grained regime was quite original. Actually, I was a bit upset that they didn't accept that in top conference because I think in that case they actually made a mistake. But that's my opinion. Anyway, we had quite some discussions here about the role of the prior. And you argue that indeed, in some situations for large data sets, the prior is actually not that important because the data overwhelms it. But I also think that in your thesis, there are situations, and also in neuroscience in general, there are situations in which this is not the case, regardless of the amount of data.
And in that case, the dynamics of the prior actually makes a very important difference in making correct inputs. So can you maybe elaborate on this kind of how this kind of situation can happen within the context of analysis of neuroscience, and how important it is to specify the prior well within an analysis framework?
So do you have an example in mind in the neuroscientific context in which the prior is not crushed by the evidence?
I do, but I would like you to come up with it because it's in your thesis.
I guess this is the MDSI, the coupling matrix, for instance, or no? Okay.
You're right, the right direction. So
for instance, in the MDSI, you're in the presence of a large evidence, but the issue is that I would think like the bigger issue is the interrelationship between the different random variables in this case. And my strategy was to try just two priors in this case. So, and to assess over a small scale whether it changed anything or not. And I was quite impressed because in the small data regime, the choice of prior was not that important, meaning that if you had a Laplace sparsity inducing prior or uniform prior in the regime of like 3 regions or so, you were obtaining more or less the same. But the prior was becoming more and more important in the high data regime in the coupling matrix, because then the sparsity inducing, so in synthetic experiments, there was a lot of noise in the estimation of the matrix, and then the fact that we imposed the prior actually helped a lot stabilize what we obtained. So this is the regime that is closest to what you seem to imply, meaning that in the high data regime, the prior was more important than the low data regime.
Although for that specific example, if you had like an infinite amount of data, the prior will become irrelevant.
Yes.
But in the context of the same cognitive analysis, there is a set of parameters where that will not happen even for an infinitely large amount of data.
The HRF in this case, yeah. True. So in the HRF, I don't know how we proceed going on, to be honest. Right now,
can you elaborate? Why is it the case? Why are there situations sometimes in Bayesian inference in which you need the prior regardless to the where you don't have concentration, so you don't have this convergence of Bayesian and non-frequentist methods?
There's a new amount of the data. So I position this, for instance, in the spectrum. If you add more data, you always have some kind of a low pass filtering effect of your bold signal. And it means that you cannot identify certain parts of the spectrum in your latent regime, and therefore you cannot identify certain parts of the spectrum in your hemorrhagic response, I guess. That being said, I think that there would be a way to obtain a better performance in here, not through the time dimensionality, but if you consider that, for instance, your HRF was more or less stable across individual for a given part of the brain, I think you could become more confident in the HRF in the high data regime by considering that you have some kind of a perturbation modeling here. So that the location, that the HRF at a given location would be more similar across subjects, means that we could refine the estimation of the HRF even in this context, I think.
Yeah, but in this case, I mean, unless you have direct neurophysiological evidence, your confidence in the consistency of the HRF is basically part of the prior, in the sense it reflects your own confidence. It does not really reflect the strength of the evidence in the data.
But this is a way to extend the amount of evidence that is going to weigh in onto your estimation of the HRF in that context. So by having evidence across subjects in that regard. So I guess the illustration I wanted to make was that this is also because of certain independent assumptions that we make in the prior, and other assumptions could make us more confident in the HRF. And I think this actually would be the way to go, given the almost non-identifiability of the sorry, the non-identifiability of the HRF just by observing the bold. And there is evidence actually in the paper that we cite in the MDSI. They actually study the HRF across individual and across the brain, and they show that it's time to peak and presence of an initial offset. It's quite stable across an individual at the same place of the cortex. So I think it would be a good prior in this regime.
Yeah, I wasn't arguing that it's not a good prior.
Okay.
Just arguing that there are situations where you cannot rely on concentration.
Yeah.
And then actually it's very important to specify the prior correctly. Because going in this direction, I think what very interesting in the connectivity analysis is that you get a very uninformative posterior in the HRF parameters, as expected, because as you said, they're undecidable, which, however, can lead you to sometimes being undecided about connectivity, but sometimes give confident statement about the direction of connectivity. So can you elaborate in the role of marginalizations of those parameters and how kind of a Bayesian analysis can help by marginalizing some parameters?
Well, marginalization is also like, I think, linked in here to the codependence between. So like the fact that you have a large span when considering the marginal distribution of one HRF, doesn't mean that by not considering the other HRFs, so if they co-vary in certain directions, you could still have a pattern of a functional connectivity that is stable, even if your marginal is quite large in that regard. What was it? Can you repeat the last part of the question?
If you can elaborate on the importance of marginalizing what you cannot infer from the data. Because I think it's something that is often, as a statistician, we like to look under the lamppost. We like to have our theory in places in which we know that we can have guarantees, and that in auto-scientific situation that actually does not happen, and that is not the case. And then we stop having much theoretical guidance.
Yeah.
Which I think is a bit of kind of the insights of your paper about connectivity. So if you can elaborate on the role of using variational inference for marginalizing sorts of uncertainty, even if you cannot estimate them directly.
Yeah, well, this is basically what we do in here. So like the HRF is not identifiable, but the fact that we jointly sample it as part of the joint posterior means that we effectively marginalize it when we sample the directional coupling. And I think it's really what matters here. So I would even argue that we go a bit one step beyond that. And the fact that we separate those hyperparameters from the parameters is a very generic methodology. Well, as you know, in your value approach. And like the fact that I would go one point beyond is that if you think that something is not identifiable or hardly identifiable, I would argue that especially for second order statistics or things like that, you are at a high risk of mode collapse. And using the forward KL in this regime, and then re-exploiting this module in the reverse KL where you think you have identifiability, conditional with the hyperparameters, is something that I think is way more generic than that. But I don't know if I'm making sense. Or well, anyway, so I'm quite convinced that marginalizing the things that we don't know, even if we cannot identify them, is important, and is one of the good advantages of rational inference, because some parts of your posterior may not converge, that you will still sample from a very large support. But in this case, we could almost sample from the prior and we would have the same thing. This is a bit the thing I'm not quite convinced about our approach because the span of the HRF angle is so large. Then we could just marginalize the prior and obtain kind of the similar result.
So do you think that is the case? In this case, you think that it's not even needed to try to evaluate the posterior over those parameters?
No, I think it's super important to be conditional on them and to effectively marginalize them. I'm just saying that in this context, oh, sorry. So in this context, I'm not sure we would obtain much difference compared to marginalizing the prior.
But this is because like we take a very large range for the prior, and actually in the range that we use over the HCP data, for instance, the prior is much narrower. It's like twice as more narrow, and the identifiability is higher, and it makes more sense. Yeah, it's quite large.
Yeah, but I mean, I think it's the advantage of automatic variational inference, and the way you present it is that sometimes your posterior is the prior, and then what? Better you're able to cover the prior, and then that's just so it's not that you need to think as a statistician, is it going to be useful or not? Well, your model, if the data is uniformity, would return the prior, and that's the best you can do for me.
And then again, like automatic variational inference offers the possibility to basically do the two. Like you treat with the same framework everything. Some parameters you cannot identify, but then they will be marginalized in the estimation of the parameters you can. And I think that's the strength of this framework, like not to have to make those strong choices and to rely on prior knowledge of the problem and to just deal with everything with the same framework.
Yeah, I mean, I feel often like I get the criticism from Laplace approximation other places that, okay, but you guys are biased. But the truth is Laplace is unbiased when things concentrate, but the reality is that when things concentrate, almost everything works. And you can make actually be unbiased and provable when you are sure that things concentrate.
Yeah.
And if they don't, Laplace can give you those horrendous results that you get here in VI, and is so that the problem is lack of concentration in general. Maybe just
so your point would be that AVI works even in the context of absence of concentration, and that's the advantage is that this framework can marginalize in the absence of concentration.
The point that we need to deal with it. There are a lot of situations very useful in science, like the connectivity where concentration does not exist. And those are real situations that we would like to be able to prove things, but we can't. And I think that you have a very nice approach for dealing with this kind of situation. So that's, yeah, so that's yeah, thank you. Okay, maybe a very last question. We discussed before the structure of conditional dependencies, and you mentioned faithful inversion. And do you think it is enough? Do you think that is because, of course, in the cascading flow approach, you have that you're not only respecting the conditional dependencies, but you're also embedding the actual functional form. Do you think it is enough in the context of variational inference to simply put the conditional dependency structure in? Or do you think that it's also important to have the specific functional form of the model within your variational posterior?
I think it's much like to the notion of initialization, like starting from an existing functional form may be useful. That being said, so I have 2 points on this. So the first one is that the functional form is a good first approximation, I agree. But in my experiments, I don't know if it was the case because you were using your highway flows architecture and cascading flows, but this created a lot of uncertainty to have a moving base distribution applied as a basis of a flow. And so today I would be much more convinced to having a stable base distribution and then just to have the conditional flow on top of it. And the better answer would be that if the problem is initialization, I would proceed with, for instance, considering different order of statistics sequentially. So like you could fit your first order using a maximum a posteriori, then you could fit your variance, and then you would go to the higher order statistics by adding a flow on top of that. And in my experiments, this was performing very well, and we didn't need any initialization using the functional form. But you had still full conditional dependencies. It's just that they were not happening at the level of the base distribution of the flow, but of the conditional transformation. If I had the time, I think I would have tried to do at least a methods paper or like a workshop paper on this kind of architectures. And in this context, yes, I think that doing this kind of faithful inversion of the template, even into stochastic regime, and fitting sequentially your statistics for your posterior would be, I think, the most computationally efficient. I had preliminary experiments for this, but never quite at the time.
So in my experience, this works when you have a linear model, like in the connectivity case and like in the hierarchical case. But for strongly nonlinear models, like often with chaotic behavior, the network is incapable of learning the correct dynamics, even if the network potentially could. So we have like situation in our paper, for example, in which the flow is completely incapable to learning the Lorentz dynamics, although it's well within its range. And then if you don't put it in, it's the model is simply incapable of learning it.
Okay, interesting.
So I think that some of the troubles you actually had was that, for example, in the connectivity semantic, you get that your prior was unstable because you needed to constrain the coupling matrix to actually be within the range of stability. So that's
yes.
Yeah, and so that's of course difficult because then it means you're taking your prior very seriously, and being a high multivariate linear model, it's actually there is a lot of stability constraints. So that's but there are situations in which if you have a complex nonlinear model, like sometimes models you would use in physics, in my experience, the network fails to learn them entirely in a lot of situations.
If you don't use the functional form of the prior.
Exactly, at least with forward KL, probably not with reverse KL. So maybe if you go with reverse KL, then it's a different story. But with forward KL, for some reason, which are not obvious to me, often it fails entirely. So for that, you need to be a bit careful.
I think Pedro, you do something like that, no? At some point, looking into base distributions and switching them depending on situation.
I was curious about it.
Okay. I think, like, for instance, in strongly multimodal with like modes very far apart, your flow will invest a lot of energy trying to like push away the density so I can perfectly imagine that in some cases you fail if you don't have a close enough functional form to the distribution.
Yeah, often, for example, in time series, the model spend a lot of energy modeling the local structure, but then it gets the global entirely wrong. So that's very common and something. But again, these models are linear, so in this case, it doesn't really matter. Okay, but anyway, thank you very much for your analysis.
All right. So the last examiner is Pedro Rodriguez.
So thanks, Reed, for the presentation. It was really interesting and clear, and all the images and pictorials are really good. There were many questions already, so I'll try to not pose too many long ones. The first one that I was curious about, like, for the PAVI and David, all your contributions, there are many bricks, right? There is a part of amortization. There's also the RKL, the forward KL, expressively of normalizing flows. So there are many parts that come up to become this contribution. I was just like curious of how important it is to use actually a normalizing flow with all the flexibility that it has. If you could use, for instance, in your neuroscience results that you had in the end, if you had used just Gaussian mixture as your density estimator, would have been like chaos with very poor results, or how did you check this kind of stuff?
As a matter of fact, we tried. So the general idea is like to amortize density approximator across repeated structures. We try to do so with Gaussian, for instance. The mixture of Gaussian, I never tried to do, but I think
I know Florence, is it? Like, that's kind of like a thing. So, I know this is a very expressive kind of family, and honestly, I would be surprised if there are not like many cases that can reproduce the same principle of the Pavia architecture, but replace flows. So that being said, flows, they have one important distinction compared to any density approximation is that it's dysfunctional form. So like in Pavia architecture, we kind of tied to the flows in the sense that we push forward the generative model distribution and not just any distribution. So you could use any other type of density approximator in that context, but you would not benefit from the same nice properties, which is then again that you have a good starting point for your flow in terms of the distribution you need to approximate.
Okay, thanks. Another question for slide 87 that you were talking about effective connectivity and stuff like that, like for neuroscience. I'm not a specialist on that, but whenever I see these keywords, it makes me think about dynamic causal modeling, DCM.
Yes.
Which I'm not particularly a fan either because I never really understood your papers, etc. But I just was curious to know if there was any connection to the stuff that you did. And,
there is a massive connection with on DCM. As a matter of fact, the model they used on the line is very similar to ours, just transposed into the frequency regime in the later iterations that they did. So Kressel et al. In 2017, they did this regression DCM, which is the newer and improved version and scalable version of DCM. And they use a variational method to infer much similarly to what we do the coupling matrix. The major differences is that they don't have the same conditional dependencies, for instance, in the posterior for the matrix. They are in the frequency regime. Last but not least, they do consider that the HRF is fixed as the default HRF. And so, for instance, in this methods paper that we're building with Luca, want to submit at the next NeurIPS, you can see here the coverage of the ground truth through the posterior on a synthetic example. So in a synthetic example, that does not necessarily have that the HRF is the default HRF in every region. So you can see that RDCM fails because it has the wrong prior in here. But the thing is that if you try to do naively the taking into account the HRF variability, which would be like the case with the reverse scale baseline that I presented in the presentation, you also fail because sometimes you end up into the good local minimum, sometimes you don't. And our method, so in expectation, has a much better coverage of the ground truth, and that can be extended to large-scale networks such as 91 degrees, where you can see that we can, with a slightly higher but on par, even in this context where the HRF is not variable, so we are on par with RDCM. So yes, it is completely compatible. I think that the next step in their papers, because of the discussion, is that they consider that the HRF variability being absent is one of the main limitations of the work. So I think they would like to go there. And therefore, there could be a completely simple combinations, which is to apply our method, but in the frequency regime, to correctly marginalize the HRF. But yeah, the confusion is there. And as a matter of fact, I know that we talked about this here a long time ago. So this is the output of all this.
So this is very interesting, right? Because in DCM, they always been using this forward KL more or less for optimizing. So, and they have also this complex observation model. They used to have this balloon model, right? With many parameters, probably undecidable. So based on your results, can you can you trust that kind of analysis?
I didn't know that the parameters in the balloon model are optimized in the RDCM. I know that they are in the original DCM. Yeah. No, I wouldn't trust those models. I think most likely they must fall into. Okay, that's just my advice, my opinion on that. I would be very touchy about any. But switching to a forward KL would
prevent that. And I think it would be a much better method to do such a hybrid inference method to try to identify the balloon Wien-Kessel parameters along with the actual coupling. The issue is, I think, with like Bayesian methodology in general, is that because you obtain a distribution, you think that you have captured all the uncertainty. And so you see a distribution, you're like, okay, good. But the distribution of distributions is quite large.
Okay, so my last question is like all the applications that you showed in your manuscript that you did not include here all of them for today, they are really challenging, like many big data stuff that people could spend the whole year working on. So you did a lot of stuff that was really impressive. And however, like there are many methodological contributions such as amortization, I mentioned, the expressive, etc., all the plates and templates, etc., that you described in the manuscript. I would just be curious to know, like, could you imagine, like, propose other kinds of applications apart in neuroscience for methodology that you have proposed, such like, I don't know, other ways of instead of just fixing with the neuroscience, not necessarily talk about sums as you did in your examples in manuscript, like,
yeah. So I think MDSI has a general application on time-shifted signals beyond just the action functionals. Like in the realm of neuroscience, we could apply it to many different things. For instance, calcium imaging also has this problem of time shifts and things like that. I think the architecture that has the most potential for applications in general is the one microstructure estimation. I liked it a lot, and I think in many cases you can have a simple local simulator of what is happening that is well suited to SBI, but then you want to move on to multivariate analysis, typically by comparing many different units. And in this case, you cannot just apply the SBI methodology because even the space for amortization is just too big. And then considering perturbation using the reverse KL of these forward KL elements that you built at the unit level, I think could be applied to, so I don't know, imagine physics or things like that. Because you have a very good model of your local dynamics, and then you want to entangle different units. And, yeah. But I don't have identified.
So you spent already enough time on the other application. So thanks.
All right, thank you very much. I'll just step into the member of the jury for 2 minutes and ask you one question, maybe two, and then I'll let Damien say a few words. So, you know, I've been teasing you for many years on many occasions, and I'll continue. So where is the story about the salmon coming from in your thesis and the fact that there's a budget cut for the scale?
Who laughed at the salmon joke in the thesis document? Well, okay.
Who paid attention?
Actually, it's not self-evident, I think. So
is there a reference to the dead salmon in the fMRI literature?
No, there's no reference. But it was quite hard to actually come up with something where the illustration of the bias would like happen. And so I like this budget cut because I'm peaking. But
okay. So just overall, I'd like to congratulate you for the clarity of the presentation, and I think you made an excellent job in your presentation and also the manuscript. There's a lot of insights showing that you've understood many things over the last years, and I guess I will stop here and let Damien say a few words, and probably Damien will also say good things.
Okay, I'm not going to ask questions because I've done it for 3 years. Maybe most of your family is more comfortable with French, so maybe switch to French. Okay, je vais faire ça en mon français avec un accent un peu maladroit. Mais en tout cas, c'est un peu la fin de l'aventure avec lui. Où on a commencé en passant différentes étapes avec différentes difficultés à chaque étape.
Ouais.
Des tout petits, mais à chaque fois réussite. Et je pense qu'une des choses à remarquer plus de lui, c'est pas seulement sa grande qualité comme chercheur et dans la partie mathématique, mais aussi sa grande intelligence émotionnelle et sa façon d'arriver à être partie d'une équipe, mais aussi d'arriver à booster les gens autour de lui et que ça arrive à se mettre en place depuis le rôle qu'il a pris comme étudiant au début jusqu'à la fin, quand il a pris un rôle senior en train de l'idée des projets et de coacher les gens autour de lui. Et en tout cas, je pense qu'il arrive à être un membre très, très valorisé de l'ensemble Mind et que ça va à se sentir en fond de son départ. Et ça, c'est tout pour moi. And now it's,
Okay, so now we'll stay in this room and we invite on va laisser tous les gens de l'auditoire nous laisser quelques minutes pour écrire le rapport de thèse.
Quelqu'un qui est unable connected to the Zoom.
Et voilà, je pense que vous pouvez compter 10 minutes.
La breakout
Dégourdir les jambes, faire une pause physiologique et puis.
Donc du coup, je fais une breakout room.
Oui.
Je vais là.
Non, t'inquiète. Parce qu'il y a les...
Si, ouais.
Ok.
Si.
Hello, Ruby. Yes. Okay. Yes. We're going to like move into a breakout room so we can deliver we can discuss about the presentation and then come to the conclusion. Yeah, for 5 minutes. So you stay with us. We
Okay.
discussed what we're going to say about him and whether the presentation was up to level. Now people are leaving the room such that we can discuss in private. Yeah.
Right. Okay. Yeah.
Thank you so much for staying with us.
Yeah. No problem.
Alex, je te laisse la parole. J'y vais faire un poste parce que moi, je ne peux pas discuter non plus. Je vais faire un poste physiologique.
Tu peux faire un test, parle pour voir si on entend bien.
Mais en fait, surtout, elle n'a pas rejoint le breakout.
Il faut l'inviter manuellement.
Je peux la sign to manuellement. Bref, hop là. Parce que du coup, ouais, pour contester ce truc est sur YouTube, donc je veux pas que la délibération soit sur YouTube.
Okay, perfect.
So I'll leave my laptop with Alexandre. I'll leave my phone.
All right. Thanks for staying, I guess, late a bit for you. So the way it goes is that there's a report that we need to write. I sent you the email, so you've probably seen it. It's in French. I guess you can use any translation system to get a sense of this. Okay. So disclaimer, it was not written with ChatGPT. It might be edited now with ChatGPT. I don't know. No, so more seriously, wait.
Hein? Ok. Now here. There, hop.
On va se mettre comment? Se mettre sur le
Les garanties techniques, on a besoin de garanties.
Ouais.
Yeah, I mean, I don't know what Ruby is looking for, if she can look at Louis or not, or we got.
Louis, mets-toi ici, on va se mettre là.
Est-ce que
quelqu'un
vous êtes
peut
sorti de la
allumer
breakout room ou pas?
The lights, do you remember the lights on top?
Moi, je peux le quitter, non?
I don't know, I mean, it will be good if Ruby can also watch.
Yes, but if you sit here, she will be able to see, I think.
Yeah.
Just because Louis' camera is still working.
Okay.
Maybe you can
Je
stop
pense que-
sharing the slide.
Ruby? Ruby, can you hear me?
Yeah.
She can.
Vous avez le micro?
Yeah, Alex had the microphone and he took it to the outside.
Tu veux un autre micro? Non?
Oh.
Can we have all the jury members on the same side? So they feel less alone. Yeah, just like so. It feels like it's a collective.
Il y a quand même un serment maintenant. Donc,
Oh yeah, you have to do
faut
the
être un
thermal.
peu-
All right. So it's a very formal moment. So we'll try to be
Can so like people
Serious
see on the
moments.
camera too.
have to be here.
Yeah, so people can see you on the camera too. Yeah.
Okay. All right. So, être en français, le rapport est en français. Donc, Louis, tu nous as présenté ce jour tes travaux de thèse sur l'inférence statistique variationnelle pour l'analyse des données neuroimageries de très grande taille. Le jury a unanimement apprécié la qualité pédagogique de ton exposé et noté l'excellente qualité des illustrations. L'exposé, contenant une sélection judicieusement choisie de tes contributions, présentées dans le manuscrit, a premièrement couvert le contexte applicatif de ton travail, utilisant des données d'IRM fonctionnelles et en particulier les mesures de connectivité dérivées de tels signaux cérébraux. Tu as par la suite détaillé les deux principaux axes de tes contributions méthodologiques. Tout d'abord, tu as présenté ton utilisation de méthodes d'inférence statistique à base de simulateurs, à l'aide d'outils modernes d'apprentissage statistique, comme les flows de normalisation, et l'optimisation utilisant des données, des outils modernes de différenciation automatique. Par la suite, tu as montré également comment exploiter la structure hiérarchique de modèles bayésiens pour inverser des modèles de neurosciences comportant des millions de paramètres. Tu as répondu avec beaucoup de recul et de précision aux nombreuses questions du jury, démontrant la maîtrise des dimensions à la fois statistiques et neuroscientifiques de ton sujet, ainsi que des aspects numériques nécessaires à de nombreux résultats expérimentaux que tu as obtenus. Donc ta thèse a également mis en avant, tes réponses ont également mis en avant différentes pistes prometteuses pour les futurs travaux de recherche. Le jury souligne que ta thèse est ambitieuse à la fois sur les aspects d'apprentissage statistique, mais aussi sur les validations expérimentales, utilisant des données complexes et massives en neuroimagerie. Pour toutes ces raisons, le jury te confère le titre de docteur de l'Université Paris-Saclay dans la spécialité mathématiques et informatique et est convaincu que tu as toutes les qualités nécessaires et requises pour être un excellent chercheur ou un ancien chercheur, si toutefois tu prenais cette voie. Voilà, félicitations.
Pas d'accord. Pas d'accord.
Yes. You need to put your glasses, c'est écrit petit. Donc là, il y a une tradition, donc c'est un peu le serment d'Hippocrate du docteur des temps modernes, que je vais te laisser lire si tu le souhaites. Tu peux toujours faire le choix de ne pas le lire.
C'est vrai?
Oui.
Non, moi je le prépare ça.
Oui, alors c'est une nouveauté. Maintenant, il y a un serment sur tous les doctorats. En présence de mes pairs, parvenu à l'issue de mon doctorat en sciences du traitement du signal et des images, ayant ainsi pratiqué, dans ma quête du savoir, l'exercice d'une recherche scientifique exigeante, en cultivant la rigueur intellectuelle, la réflexivité éthique et dans le respect des principes de l'intégrité scientifique, je m'engage, pour ce qui dépendra de moi dans la suite de ma carrière professionnelle, quel que soit le secteur ou le domaine d'activité, à maintenir une conduite intègre dans mon rapport au savoir, mes méthodes et mes résultats. Et voilà. Du coup, en me tenant sur, so this part of the thanks is actually in English, so sorry Ruby and Luca, but so that my family can understand it. Du coup, c'est les remerciements. Je voudrais tout d'abord remercier le jury, déjà, d'avoir accepté d'être présent aujourd'hui pour me challenger avec bienveillance, bien sûr. Si vous avez trouvé le temps long dans cette présentation, dites-vous que le jury a en plus dû lire 200 pages qui sont rentrées dans tous les détails, avec des images, certes, mais bon. Merci pour votre temps, que je sais précieux, et de m'avoir laissé la chance de vous présenter le résultat de 4 années de travail. Résultat, donc, à ma surprise, je suis assez fier. So thank you, Ruby and Luca, for your precious time and letting me present to you my work, my pride and joy. Ensuite, je voudrais remercier Demian. La relation entre un superviseur et un thésard est toujours complexe. Et beaucoup dans cette salle ont sans doute déjà eu un manager, mais je pense que la relation au superviseur elle est beaucoup plus subtile. Il faut trouver une difficile balance entre chef, mentor et collaborateur, et une forme de compagnonnage intellectuel, à ma pensée. J'ai vraiment vécu un vrai tournant dans notre relation sur la 3e année de thèse, avec la participation à certaines de tes collaborations de longue date que je sais précieuses. Et je voulais te remercier pour la confiance accordée et ta supervision qui m'a fait passer de jeune scientifique idéaliste à vieux cynique dépité. Non, mais plus sérieusement, grâce à toi, j'ai pris conscience dans cette thèse de la force du compromis, d'à quel point le temps est précieux, et surtout de la dure nécessité de converger, même si ce n'est pas parfait, au terme de périodes d'exploration. Et tu sais mieux que personne que ça a été une longue route tortueuse. Donc, merci. Merci beaucoup. Plus généralement, je voudrais remercier Parietal puis Mind, qui sont 2 équipes dans lesquelles je me suis senti comme à la maison. Je mesure le temps passé à tout organiser, tout financer pour que les jeunes puissent se concentrer sur la technique, tant qu'il peut parfois être invisible depuis l'intérieur du cocon. Et je sais qu'une équipe qui combine une exigence intellectuelle et une vraie bienveillance, c'est quelque chose de rare, de difficile à construire et à maintenir. Donc, merci au PI. Et aussi merci à ceux qui triment jour après jour sous le joug des PI, les générations de doctorants que j'ai vu passer, pré- et post-Covid. Et je trouve que l'ambiance est très bonne, et ça non plus, ce n'est pas le cas partout, donc bravo à nous, en fait. Je voudrais aussi remercier ma famille et mes amis d'avoir été présents pour cet événement. 4 ans, c'est long. Et plus qu'une thèse, j'ai l'impression que c'est tout un chapitre de ma vie qui s'achève. Et je suis très heureux de pouvoir faire ça avec vous tous. C'est assez émouvant de rassembler des gens qui viennent d'horizons et de circonstances variées. Ça fait mesurer le chemin de parcours. Enfin, et avant toute chose, je voudrais remercier celles sans qui rien de tout ça n'aurait été possible. Et je dis ça sérieusement, parce qu'il en faut de l'énergie pour supporter mes angoisses et ma légère tendance à l'exagération, d'aucuns diraient au drama, du style « ça y est, Soso, c'est la fin de la thèse, le truc marche juste pas et tout le monde va enfin se rendre compte que je suis une fraude. » Puis le lendemain, « ah non, en fait, ça marche. » D'ailleurs, petit, ma mère me surnommait Calimero. Merci donc au véritable dernier auteur de cette thèse, Sophie. Et maintenant, le tour. So, thanks, Ruby, for being present.
No problem. Congratulations.
I know it's late for you. I was very happy to be able to have you from so far away. And
Yeah, right. Yeah. Congratulations again. Yes.
thanks.
It's a long journey. You made it. Okay.
Une photo? Ouais, ok. Donc là, il n'y a pas eu assez de photos, mais d'expérience. Ces photos, elles ressortent après. Vaut mieux avoir les photos de maintenant que ce soir à minuit.
Ça
va.
tient un truc.
Les gars techniques.
Je peux prendre le truc comme ça pour avoir Ruby aussi.
Yeah.
Tac.
Is there anything else? Oh, okay.
Yeah,
I see.
You're in the photo.
Okay. I don't know there's so many audience.
Qui est-ce qui prend la photo?
Les gars techniques.
Un appareil photo de merde.
Ah,
Je peux prendre la photo, mais.
Yeah.
Tiens.
That's also. They make her full screen.
Sure.
Okay. Yeah. Thank you, guys. Yes.
I don't know how to.
but you're not on the picture yet.
Ah, okay.
Je sais pas comment la mettre full screen.
Okay, c'est
Bon, allez, c'est pas grave.
Hold me in a nicer way. Yeah. Thank you.
Voilà.
Thanks, Ruby.
Thanks. Yes. Congrats.